Hello everyone, I am Aurelien Pelissier from IBM Research on ETH Zurich, and I will be presenting identifying convergent epitope reactivity in B-cell repertoire data. So epitope reactivity convergence is a key concept in immunology and in computational biology in general, where some antibodies or B-cells from different founder cells, they will evolve through mutations and selections through binding a specific antigen. And this specificity is made through this what we call the interface here between the antibody and antigen, where there is some a good overlapping of the shape, which makes a good binding. And this diversification of the antibody or the B-cell receptor comes from two different processes. The first one is VDJ recombination during the creation of the B-cell receptor. And the second one is a somatic hypermutation, which is like accelerated mutation that happens later on during the selection phase, like in the germinal center. Um, the binding is mainly comes from what we call the complementary determining region, CDR1, 2, 3. So there are 3 CDR in the antibody heavy chain and 3 CDR in the light chain. And these usually are the key factors in the binding affinity of the antibody through a specific antigen, because there are some very special loops that can take a lot of different shapes. So let's say we have this problem where we have an antibody and we want to know about his binding properties. So we could try to predict like the affinity to a given epitope, or we could try to predict the effect of one mutation of the affinity. But let's take a very simple task, like given two antibodies, do they bind the same epitope? So this task, of course, is less powerful than the other two I proposed, but you can still get a lot of information in B-cell reported data by knowing the number of antibodies binding the same epitope, for example. So to approach this problem, you can use several approaches. The first one is simply a sequence-based approach, where you're just going to compare the CDR sequence similarity through the Levenstein distance. You can also try to predict some paratope. So there is some tools that can predict accurately the paratope residue in the CDRs, and you can compare them. And finally, you can use structural information. So you can infer the structure from the sequence, which work decently well for CDRs, except for the CDR3 of the heavy chain. And you can look at the conformation of these loops and see how they match each other. So these three methods, like the method 2 and 3, they are described in literature already, and the first one can be used as a baseline. But basically, we tested this approach on the dataset where the ground truth was known. Like we know already, like it was tested with ELISA if the antibody is a binder or not of the Petruxis toxin binder. So we validated and optimized these three methods on this dataset. And what we can do with this, once it's ready, we can do the following. We can estimate the number of epitope in a sample. So once you know if a B cell is binding the same epitope another B cell, you can group them into clusters and estimate the number of epitope in the sample. This can be useful. Another thing we can do is that for special diseases that have stereotyped epitope, where the binders are already known. So let's say you have a list of known binders to some specific epitope. You can use this method to see if some of the B cell in your dataset have a similar, will bind the same epitope as the binders in this list. So this, for example, what we can do with rheumatoid arthritis, where we have a lot of known binders to some epitope already. And this is in collaboration with Amsterdam experimentalist. Finally, another thing you can do, you can identify common epitope reactivity across samples in a germinal center, or for example, in any other kind of data set that you'll be interested. But in germinal center, because the two germinal centers are quite independent from each other, two independent environments. So if we can find some antibody that came from different germinal centers that they bind a similar epitope, this could be very interesting to see. So this is what we did here at the third application. So this was just to give you an overview. Um, Later, we can test this prediction with ELISA assay, and we could incorporate, because right now we only focus on the similarities of two antibodies, but we could incorporate epitope information into prediction. And yeah, so I would like to thank you for your attention. If you have any more questions, you don't hesitate to ask me directly or during the poster session. And I would like to thank, of course, the three experimentalists that helped me through this project, and my supervisor, IBM Research, Maya Rodriguez Martinez. So have a good day, and see you soon.